My name is Jasmine, and me and my partner Aisha today will be hopefully covering the entire chapter of oxidative phosphorylation in 2 hours. So, inshallah, we can do that. So, let's get started. Okay. Ah. So, basically, this is the total spectrum of concepts that we will be covering today. Basically, everything that's included. And I just want you guys to note that after every section, we're going to be testing your knowledge with Kahoot. So do keep a spare device in hand so that you can join us. Okay? Can you guys hear me?
Yeah.
Yes.
Okay. So before we get into this topic, let's just have a general idea of what happened in the three processes before it. So in glycolysis, we broke down one molecule of glucose to form 2 molecules of pyruvate, 2 molecules of ATP, and 2 molecules of NADH. Now, each molecule of pyruvate is going to enter the link reaction, where it's broken down into acetyl-CoA and NADH, right? So, but remember that because we form 2 molecules of pyruvate and each one will enter the link reaction, then we're going to get 2 molecules of acetyl-CoA and 2 molecules of You guys can hear me or no?
Yes.
Yeah.
Okay. Because it shows that I'm muted. So because we got 2 molecules of pyruvate, then each one will enter the link reaction and form 2 molecules of acetyl-CoA and 2 molecules of NADH. Now, each molecule of acetyl-CoA will enter the TCA cycle, where it forms 3 molecules of NADH to a molecule of FADH2 and ATP. Now, same story, because we form 2 molecules of acetyl-CoA, we're going to get 6 molecules of NADH, 2 molecules of FADH2, and 2 molecules of ATP. So now if we add up the total, I put this table as a summary of basically what I said. So if we add up the total, we will get 2 plus 2 is 4 molecules of ATP, and 2 plus 2 plus 6 is 10 molecules of NADH and 2 molecules of FADH2 before oxidative phosphorylation. Now, what you're going to notice is that during oxidative phosphorylation, NADH and FADH2 will be converted to form more molecules of ATP. So this is going to become 0, and this is going to become also 0, and this is going to increase in number. Okay? So you know that there are 2 types of electrical charges in this world, right? A positive charge and a negative charge. And now when we bring these 2 charges together, what's going to happen is that they're going to attract each other. Now this attraction is called affinity. So you could say that they attract each other, or you could say that they have affinity towards each other. Now if because of this attraction or affinity, they actually come together and bind together, that's called reduction. So the ability to do that is reduction potential. Okay? So if you have, for example, 2 molecules, A and B, and let's say that A has a higher reduction potential than B, then A is more likely to steal an electron from B. Why? Because A is more able to or A attracts the electron stronger than B, if that makes sense. Are you guys following so far?
Yes.
Okay. And this entire process, by the way, this entire process releases energy. So you should know that. Okay, so next. So I have a question. You know that aerobic metabolism consists of TCA cycle, the link reaction, and oxidative phosphorylation. But you've seen that neither the TCA cycle nor the link reaction use any form of oxygen. So then how come we call them aerobic? This is because the last process the oxidative phosphorylation, requires oxygen, and it's kind of in the name. But also then, okay, that's fine. So then, but then like, why oxygen? Like, why specifically oxygen? Why doesn't it require sulfur or like nitrogen or like any other atom in the universe? Why oxygen? Does anyone like have an idea of like what the benefits of like this process requiring oxygen and not something else is to us?
Because oxygen has the highest reduction potential?
In the ETC, yeah.
And when it gets reduced, it's just going to be water. It's not something that's foreign or toxic.
That's true, but also it can, when it gets reduced, it can form something called reactive oxygen species, which are, we'll get to that later, but basically they're harmful substances to the cell and they can cause cancer. But مثلا, for example, one advantage of this is that oxygen, you can find oxygen anywhere. So we need oxygen for this process. So it's not like, oh, if I go to a certain like country or a certain place on like the planet, that I'm not gonna find oxygen. No, like if that makes sense, like no matter where I am, I'll always have oxygen available. So for me to need oxygen for this process and not something else, that's a benefit for me because it's just found anywhere and I can get it anytime. Another uh, uh, another benefit of this is that oxygen is not polar. So like it doesn't require a special protein in the membrane to like cross the membrane. It can easily diffuse across the plasma membrane and it can easily enter the cells. So that's also another benefit. Another benefit is that because of its structure, so like you said, it has a higher reduction potential. That means that it can accept electrons more easily because of its structure. But the negative or disadvantage of this specific last point is that it can also form reactive oxygen species very easily. So do you guys get this point? Or would you like me to repeat?
But isn't reactive oxygen species something bad?
Yeah, that's a disadvantage of that point.
Oh, okay.
Yeah. So like these three are like advantages, but this is just a disadvantage, if that makes sense. Okay, can I move on? Whether you have questions.
What is a diradical?
That means it can have two electrons.
Okay, thanks.
Like because of its structure, it has like basically you can say it has the two empty spots and each empty spot accepts an electron. So we say diradical. It means it can accept two electrons. Do you get it?
Yeah.
Okay. Anything else? Okay, I'm gonna move on. Okay, so now, like I've mentioned it a couple of times, like ETC, ETC, ETC. What is ETC and what is the electron transport chain? Is it the same as oxidative phosphorylation? No, the ETC is a part of oxidative phosphorylation, whereas oxidative phosphorylation is like an entire process that basically it takes electrons, okay, it receives electrons and it moves them throughout a set of 4 proteins that are found in the inner membrane. These 4 proteins are called the ETC, or the electron transport chain. And basically what happens is that electrons move from one protein to another. So because they're moving from one protein to another, we call these proteins electron carriers, okay? Because they're like basically carrying electrons, like they receive it and then they donate it to the next. And In order for one protein to accept electrons from the other, it has to have a higher reduction potential than the other, right? Like I mentioned before, it should be able to steal the electron better. That means that these proteins or carriers are arranged in an order of increasing reduction potential. And this whole process by which the electrons are moving from one carrier to another, it releases energy. And this energy that is released is used to synthesize ATP, okay? And that process is called ATP synthesis. And so ATP synthesis and the electron transport chain collectively form the oxidative phosphorylation process. But, okay. But then, like, where do the carriers often get the electrons from in the first place? Like, do they get electrons just from anywhere? No. They receive electrons from coenzymes called NADH and FADH2. So remember when I told you previously that NADH and FADH2 are converted to form ATP? This is how. Because NADH and FADH2 will donate their electrons to the electron transport chain, which is used to which moves around and releases energy. Okay, and this energy is used to synthesize ATP. But okay, then when they keep moving around the carriers, like, do they just keep moving around, or like, do they have like a final destination? Like, do they stop somewhere? Yeah, they do. They stop at oxygen. So this is why we need oxygen for oxidative phosphorylation to occur. So therefore, since oxygen is the final electron acceptor, we can say it has the highest reduction potential. And because the coenzymes NADH and FADH2 are the initial donators, then we say they have the lowest reduction potential. Does this make sense?
Yeah.
Yes, it does.
Okay. So then, like, okay, they release energy when they're moving around through the carriers, but is this energy only used to synthesize ATP? No, it can be used for other processes, like what? مثلا, in certain tissues called brown adipose tissues, we can use this energy released to generate heat. For example, these tissues are very much abundant in babies. So, like, I think when you carry a baby, like, you'll feel that the baby is warm. That's why, because they have a lot of brown adipose tissue, which generates heat by the energy released during electron transfer. But another purpose that they could be used for is to pump calcium into the matrix. Why would we need to pump calcium into the matrix? Remember in the TCA cycle when we said that calcium activates the certain enzymes of the TCA cycle? And remember that the TCA cycle occurs in the matrix? This is exactly like linking it. Like, calcium can be pumped into the matrix to activate the TCA cycle. How? Because of energy release during electron transfer in the ETC. Okay? Next.
Can you repeat that last part, the calcium part?
Basically, so remember how in the TCA cycle, we mentioned that it could be activated by calcium? Calcium is an activator of the TCA cycle, and the TCA cycle occurs in the matrix. We're just explaining how we even like put calcium into the matrix so that it can activate the TCA cycle. It's because of the energy released during electron transfer. I don't know if that makes sense.
Yeah, thank you.
Okay, great. So, okay, so now that we've covered the fundamentals of like ETC in the background and everything, let's go into detail of each complexes. Remember, there were 4 complexes, okay? Or 4 carriers, 4 proteins, whatever you want to call them. We can also call them oxidoreductase enzymes. What does that mean? It literally means electron carriers. Why? Because oxido, so oxidation, and then reductase, so reduction. So basically, that's the definition of electron carrying, right? So we have 4 complexes. First one is called NAD dehydrogenase. The second one is called succinate dehydrogenase. The third one is called cytochrome BC1. And the fourth one is called cytochrome C oxidase. Okay? So just an overview of what they do. Complex 1 is going to receive electrons from NADH. And it basically sends those electrons to complex 3. Now notice that it doesn't send them to complex 2. Complex 1 is actually independent of complex 2. Okay? How? Because complex 2 receives electrons from FADH2. Okay? So it doesn't receive electrons from NADH. That's only complex 1. And complex 1 doesn't receive electrons from FADH2. It only receives it from NADH. So basically, complex 1 and complex 2 are independent of each other. They're not connected. And they both send electrons. So they both receive electrons from specific coenzymes, and they're going to send them to complex 3. Okay, and complex 3 is going to receive these electrons, and it's going to send them to complex 4. So how do they send them? Is it just free floating? No. Basically, complex 1 is going to receive the electrons from NADH, and it's going to send them to a carrier. This carrier is called ubiquinol. Okay, or you could call it complex Q. And basically, I just wanted to mention, it's UQ is basically supposed to be in the inner mitochondrial membrane, but I just included it outside because I couldn't fit it inside. But this is wrong. Like, it should be inside the membrane, not in the matrix. Like, it shows that it's in the matrix, but it shouldn't be. But here I showed it's in the membrane. And so basically, they both receive their electrons from specific coenzymes, and they send them to UQ. Okay, now what UQ does, it's going to send the electrons to complex 3. And then complex 3 is going to receive the electrons from UQ, and it's going to send them to another carrier called cytochrome C. And cytochrome C is going to send the electrons to complex 4, which is going to send the electrons to oxygen. That's just a brief idea. We have the first complex, okay? The first complex, what does it do? It's going to receive electrons from NADH, and it's going to send the electrons to ubiquinol or complex Q. It's the same thing. Now, how does it do that? Basically, you should know complex 1 is made of 2 parts. It has a peripheral arm, اللي هو the part that's like sticking out of the mitochondria, of the membrane, like it's sticking out of the membrane. And then it has a membrane domain, which is the part that is stuck inside the membrane and doesn't stick out. So the peripheral part is the part where the electron transfer happens, okay? And what's going to happen is that NADH, from wherever source it came from, okay, is going to come and it's going to bind to complex 1, and it's going to donate 2 electrons to FMN, which is a flavoprotein, right? Now, because FMN receives 2 electrons and it's reduced to FMNH2, then each electron, they're not going to stop here. They're gonna go through 7 iron sulfur clusters. Okay. Now because complex 1 has 7 iron sulfur clusters, 6 of them are arranged in there's in each cluster. So 6 of the clusters, each of them have 4 iron atoms and 4 sulfur atoms. And then one of the clusters has 2 iron atoms and 2 sulfur atoms. Okay, so what's going to happen is that the electrons that were in FMNH2 will go one at a time throughout each iron sulfur cluster until when they reach finally their last iron sulfur cluster, they're going to both simultaneously jump from the iron sulfur cluster into complex Q, forming dihydroubiquinone. Does that make more sense? No?
Yes.
It does?
Yeah, it does make sense.
Okay. So you got the process, okay? In the end, NADH basically donated 2 electrons to complex 1, and each electron had, and the electrons had like a specific pathway that they go throughout complex 1, and they finally land in ubiquinone, forming dihydroubiquinone. That's the peripheral arm. Now, obviously, because the peripheral arm is right next to the membrane domain, the membrane domain is gonna be affected by this. And so basically the effect is that 4 channels in the membrane domain will open so that it can pump 4 protons from the matrix into the IMS. So you should know that complex 1 pumps 4 protons from the matrix into the IMS, okay? 7 iron sulfur clusters. It does contain a flavoprotein, which is FMN, and it receives electrons from NADH and donates them to ubiquinone, forming dihydroubiquinone. Does that make sense for you guys now? Do you mind reacting if that makes sense? Okay, next.
We understood.
Okay. So now we move on to complex 2. Complex 2 is also called succinate dehydrogenase. Does this ring a bell for you guys? The name? Do you remember hearing
Yes.
this anywhere? Yes. Where?
Yes.
Succinate to fumarate.
Yes, exactly. During the TCA cycle.
Step 6.
Yes. So basically, this enzyme is also involved in the TCA cycle. Do you guys want to revise the structure? Can anyone like remind me of the structure about it?
Structure of what?
Succinate dehydrogenase. It contains 4 subunits. Should A, should B, should C, should D. Okay? And should A is a flavoprotein. That means it contains FAD. And this is basically where succinate is going to be converted to fumarate, and it donates 2 electrons to FAD, converting it to FADH2. Okay? Oh, sorry. And basically, FADH2 will donate their electrons to should be. What is should be? A series of 3 iron sulfur clusters. Okay, so like complex 1 contains 7 iron sulfur clusters. Complex 2 has 3 iron sulfur clusters, and they're specifically found in should be. Okay, now does it donate them? Does FADH2 donate the electrons simultaneously or one at a time? It donates them one at a time. Just
One
like
at a time.
yes, just like complex 1, how FMNH2 donated the electrons one at a time to the iron sulfur clusters. Now the difference between complex 1 and complex 2 is that in complex 1, once the electrons finished the iron sulfur clusters, they simultaneously both get donated to ubiquinone, forming dihydroubiquinone. But in complex 2, instead of both being simultaneously donated to ubiquinone, they're donated also one at a time. So the whole process in complex 2 is one at a time. Okay? And so what's going to happen is that one electron is donated to ubiquinone, forming hydroubiquinone. And then another electron is going to be donated, forming dihydroubiquinone. Okay? Does that make sense for you now?
Yeah.
Okay. Now, remember from TCA cycle that between should C and should D, what's the function of should C and should D?
Can you repeat the last part?
Which part specifically?
Ubiquinone.
Can you repeat? Ubiquinone? Basically, in complex 1, even though FMNH2 donates the electrons one at a time to the iron-sulfur clusters, they both simultaneously are donated to ubiquinone after they finish the iron-sulfur clusters, after they go through all the iron-sulfur clusters. But in complex 2, FADH2 donates the electrons one at a time to the iron-sulfur clusters, and then once they finish going through the iron-sulfur clusters, they're still donated one at a time to ubiquinone. So first electron is donated, forming hydroubiquinone. Then the second electron will be donated, forming dihydroubiquinone. Does that make sense?
Yes.
Okay. So back to what I was saying, in should C and should D, they anchored the complex into the IMM. So they basically like supporting structures, and between them there was a heme group, right? But this heme group is not involved in electron transfer. All it does is that it prevents electrons from leaking into like the matrix or the environment in general, okay? So it prevents formation of reactive oxygen species. Also, did you guys notice something? That did you notice any protons being pumped across the inner mitochondrial membrane?
No.
So that means that complex 2 is the only complex that does not pump electrons across the inner mitochondrial membrane. You should know that. That's very important. Now,
If I can just interrupt you for a second. You said the heme group prevents leakage of electrons
yeah.
from the intermembrane space to the matrix, or من وين لوين?
No, it just prevents the leakage of electrons from, like in general, from the ETC or from complex 2 into anyone. Like
It prevents the leakage of electrons from complex 2, صح?
yeah, from complex 2 into the matrix or the intermembrane space.
And it results in O2 radical formation.
It prevents it.
Okay.
Like the heme group, because it prevents electrons from leaking, it prevents formation of reactive oxygen species. Okay?
Yes, that's clear.
Okay, great. So so now what you'll notice about complex 2 is that we have alternatives for complex 2. Not all electrons will be donated through complex 2. What are these alternatives? You have two. You have glycerate 3-phosphate dehydrogenase and you have UTF. Okay, so glycerate 3-phosphate dehydrogenase, we'll go through it later because it's actually like the process. It's actually involved in a process called the glycerate 3-phosphate shuttle. But we can go through UTF for now. So UTF is basically an enzyme that will receive electrons from like enzymes in the fatty acid oxidation and amino acid oxidation. So not carbohydrates metabolism. Okay, so for example, in when fatty acids or amino acids are oxidized, you learn this later. Basically, they can get oxidized, and the products or the enzymes will go to UTF to donate their electrons. Okay, And UTF will receive these electrons and they will donate it to UTFQO, okay? Which will directly donate the electrons to ubiquinone, forming dihydroubiquinone. All good?
I didn't understand.
Which part?
From glycerate to the UCF.
Can you repeat? I couldn't hear you.
From the glycerate to UTF.
It's not from glycerate. It's basically any oxidation of fatty acids or amino acids will donate their electrons to UTF, which donates the electrons to UTFQO, which will donate the electrons to ubiquinone, forming dihydroubiquinone. Glycerate 3-phosphate is separate.
In the book it says ETF. Is it the same as UTF?
Ah, I must have spelled it wrong. Sorry.
That's why I was confused. Thank you.
But is it clear now? I'll fix this mistake when I send you guys the PowerPoint. But thank you for pointing that out. Is that clear?
Okay. Yes.
Okay, so now complex 3 is a bit complicated. Basically, when complex 2 and complex 1 receive their electrons and donate them to ubiquinone, and then ubiquinone, where will it go? It's gonna, or dihydroubiquinone, will go to complex 3 to donate its electrons, okay? Now, complex 3 will basically make the electrons go through something called a Q cycle. So let's first go through the structure of complex 3, and then we'll deep into Q cycle. So basically, complex 3 is a homodimer, okay? Meaning شنو؟ إنه it's made of 2 identical monomers, and each monomer contains 11 subunits, 4 of which are cofactors, okay? And these cofactors are called cytochrome Bl, cytochrome Bh, cytochrome C1, and 2Fe, 2S. So basically 2 iron, 2 sulfur cluster. So basically what it's gonna do is complex 3 is gonna receive the dihydroubiquinone, right? Now the dihydroubiquinone will come here, okay, and it will donate 2 electrons to complex 3, but not at the same time. So again, one at a time. So dihydroubiquinone will first donate 1 electron to the iron sulfur cluster, which will donate the electron to cytochrome C1, which will finally donate the electron to cytochrome C, which is the final acceptor of complex 3. So then cytochrome C will be reduced, and then you will form hydroubiquinone, right? Because we only donated 1 electron. Now, the second electron will be donated to cytochrome Bl, which donates it to cytochrome Bh. Okay. Now, the final products will be ubiquinone, reduced cytochrome C, and 2 protons, which are just like lost to the intermembrane space. Do you get this part? Well, you want me to repeat?
No, it's good.
Okay, this is the first round of the Q cycle. Now, the second round involves another dihydroubiquinone, and it's basically going to do the same thing almost. It's going to donate 1 electron to the iron sulfur cluster, which donates another electron, which donates the same electron, sorry, to cytochrome C1, which finally donates it to cytochrome C. Now the product is gonna be hydroubiquinone, which will donate another it will receive an electron. From where? I didn't include this. Sorry. I'll change the PowerPoint and add it. But basically, in المفروض from here, this one is gonna, so the cytochrome BH that was reduced in the first round will donate its electron to this hydroubiquinone, forming dihydroubiquinone again. So in the second round, only one electron is donated, while in the first round, 2 electrons are donated. Okay, and in the second round, the hydroubiquinone will receive an electron from cytochrome BH. And so therefore you'll form, this will get oxidized back to its original state, and you'll get 2 cytochrome Cs that are reduced. You'll form dihydroubiquinone, and 2 protons will be lost to the IMS. Do you want me to repeat? It's fine if you don't get it. Okay, I'll repeat.
The products?
Sorry? The products? Basically, we're gonna get 2 protons that are lost to the IMS from the first round, and another 2 protons that are lost to the IMS again in the second round. So total of 4 protons lost to the IMS. Then because we donated 1 electron to cytochrome C, we're gonna get reduced cytochrome C in both rounds. And then the first round produces ubiquinone, and then the second round produces dihydroubiquinone. And it also oxidizes the reduced cytochrome BH to form like the original one. Do you want me to repeat the whole thing?
No, it's clear.
Okay. Now, as a result of this, again, 4 protons will be pumped into the IMS. Now, you noticed that in each complex is gonna form 1 dihydroubiquinone, which donates to complex 3. That means that both of them, or like let's say each of them, will produce 1 Q cycle. You should know that. And each complex will produce 1 Q cycle. So if we were to use both complexes, we will get 2 Q cycles. Okay. Now, lastly, the complex 4. It's also called cytochrome C oxidase, and it's basically going to receive the electrons from cytochrome C, and it's going to donate the electrons to oxygen. So oxygen is the final acceptor. So how does it do that? Basically, complex 4 is also a homodimer, and each monomer contains 14 subunits. Now, the main groups that you should know is that there's copper A and copper B. So there's 2 copper A's and 1 copper B. There's cytochrome A3, and there's also cytochrome A. Okay. So what's gonna happen is that cytochrome C that was produced in complex 3 is going to donate the electrons to Cu, like the Cu center, okay? Which is basically 2 copper A atoms. And now, does it donate the electrons one at a time? No, both simultaneously. One at a time. So everything in this complex is one at a time. Now Cu center will basically donate the electrons to cytochrome A3 CuB center, which is also known as the iron copper center. Okay, and each of these centers will bind to one molecule of oxygen. Now you need 4 of these centers, or 4 of these reduced centers, to bind to oxygen so that it can form water. So that's why you need to form 4. Now remember when I said each complex will produce will result in 1 Q cycle, okay? And each Q cycle will produce 2 cytochrome Cs. So then when you have 2 cytochrome Cs, that's already 2 of these like iron copper centers that are reduced, that bind to oxygen. And then when you get 2 rounds of Q cycle, then you multiply again by 2. So that's how you get 4. So oxygen will be then reduced to water, and 4 protons will be pumped from the matrix into the IMS. Would you like me to repeat anything?
No, thank you.
Okay, and because now notice what I said, in 2 Q cycles, because of each of the complexes, so it can form 1 molecule of water, okay? That means that if we only used one of the complexes, so if we only went through 1 Q cycle, then we would only pump 2 protons across the inner mitochondrial membrane. You should keep that in mind. That's it for the complexes. Sorry.
I don't understand what exactly a Q cycle is.
So the Q cycle is just this whole process that occurs in complex 3.
Oh.
Like the electron transfer throughout complex 3 is called the Q cycle. And it's made of 2 rounds. Why? Because each round involves 1 dihydroubiquinone that enters the complex, and it transfers or donates 2 electrons, okay? And then the second round involves another dihydroubiquinone that donates 1 electron.
Yeah.
Does that make sense, or did you not understand?
No, I got it.
I didn't understand the cycle in complex 4.
Complex 1?
No, complex 4.
Oh, you want me to repeat that? Okay. So basically, complex 4 is a homodimer, okay? Meaning it contains 2 identical monomers. Each monomer contains 14 subunits, okay? Now, the important groups that you should know in each monomer is that it has 2 cytochromes, cytochrome A and cytochrome A3. It contains 3 copper ions, so 1 of them is copper B and 2 of them are copper A, okay? Now, they're arranged in specific ways. In basically cytochrome A and cytochrome A3, and the copper B will form an iron copper center. And then the 2 copper A atoms will form a Cu center. Okay, and then, so you should know these centers. Okay, so basically, in what happens in each cytochrome C that was reduced in complex 3 will come to complex 4, and it will donate 2 electrons, one at a time, to this copper-copper center. Now, each electron, one at a time, will move from the copper-copper center to the iron-copper center. Now, when an iron-copper center, when an iron-copper center gets reduced, it will bind to oxygen, and you need 4. And eventually this will form water. But for it to be able to form water, you need 4 iron-copper centers to bind to oxygen so that it can form water. Does that make sense?
Is the iron copper center the same as A3COB?
Yeah. The
Okay.
iron-copper center is made of cytochrome A3 and copper B. Do you guys have any questions? Because we finished the complexes. So this is just a summary for you guys to recap if you want to recap and look back on it later. And okay, so now we can move on to the Kahoot. Okay, so let me open the link. This one. But guys, did you understand? You understood, right? If you want me to repeat anything before I ask you the questions, please let me know. That's the pin or the link, by the way, if you guys want to join. Okay, do you want to start now? Okay, let's start. Okay, hello. How do I make this? Oh, here. Okay. Oh, do you guys want me to revise the questions? I'm sorry, I forgot. Do you guys want me to, because this one you guys like, which features do they have in common? Only complex 2 is involved in the TCA cycle, and transmembrane proteins, only complex 1 is transmembrane protein, whereas complex 2 is only like partially attached to the membrane. Okay, we can count the number of protons after this. 2 questions left, and then I'll count the number of protons with you guys. So don't worry about that. Okay, this should be good for you guys since most of you answered correctly. Okay, now last question, and then we move on to the next section. Okay, hello. So guys, did you have any problems with any of the questions? Do you have any specific questions that you didn't?
The last
Okay,
3 questions.
okay. So 1, 2, but these 2 are fine? 3, 4. This one, starting from here? Okay, so let's count. Basically, starting from NADH, when NADH is gonna enter the ETC, it's basically will result, so it starts in complex 1, right? So we're gonna pump 4 protons across complex. Now, complex 2 doesn't pump anything, and complex 3 will pump 4 protons per Q cycle. So, for example, if we said one NADH enters the ETC, it will result in 4 protons being pumped from complex 1, 4 protons being pumped from complex 3, and only 2 protons being pumped from complex 4. Why? Because we need 2 Q cycles to pump 4 protons in complex 4. Okay? But because we only have one NADH, then we're gonna get only 1 Q cycle and 2 protons pumped across complex 4. So if we add them together, we're gonna get 4 plus 4 plus 2, which is 10. Is that clear? Or you want me to repeat?
It says 4 proton because we need 2 cytochrome C, right?
Sorry?
You said it's 2 proton, but here it's written 4 proton because we require 2 cytochrome C.
Yeah, we pump 4 protons when it's 2 Q cycles. But for each Q cycle, it's 2 protons. Okay?
Okay.
Are you sure you get it?
Yes, yes.
Okay. Now for FADH2, we're bypassing complex 1. We don't go through complex 1. So, and complex 2 doesn't pump any protons. So we're only going to have 1 Q cycle of complex 3, which pumps 4 protons. And then because we only have 1 Q cycle, 2 protons through complex 4. So 4 plus 2 is 6 protons for FADH2. That's good?
Yes.
Okay. Now, for NADH and FADH2, for both of them, when one NADH goes through complex 1, we get 4 protons pumped through complex 1. Now, FADH2 will not pump any protons through complex 2. Then we're going to get each NADH and FADH2 will result in 2 Q cycles. Okay? So we'll pump 8 protons through complex 3. And then for complex 4, because we got 2 Q cycles, we'll pump 4 protons. So then we're going to get 4 plus 0 plus 8 plus 4 is 16 protons pumped for both of them. Okay? Does that make sense or no? Moving on. It makes sense, right? Or no?
Yes.
Okay. طيب now how are they like arranged? How are the complexes arranged? We learned about the complexes, but we didn't learn about how they're like, are they stuck together or are they spaced far apart? There are two theories based on this, the fluid state theory and the solid state theory. The fluid states theory believes in each complex is spaced far apart from each other, while the solid state theory believes that all the complexes are stuck to each other, forming a like a bigger complex or a super complex called respirazole. Now, what are the differences between each theory? When the two when all the complexes are stuck together, okay, then because the electrons are going to travel a less distance to reach from electrons are going to be traveling less distance to travel from one complex to another, okay? So this results in a faster electron transfer and it's more efficient. And it also means electrons are going to be less likely to leak out and form reactive oxygen species. Why? Because like all the complexes are like concentrated in one small area. They're not spaced far apart. So that's the main point of each theory. We can go back and you guys can answer. Next. Okay, خلاص. Next section. My partner will be explaining now. So let me just...
No, خلاص, keep being presenter. I'll take it the next part, okay? Hello, guys.
Can
My
you go back to the solid state versus fluid state slide?
Sure. 1 minute. Sorry.
أنا راح طبعا.
Okay. Yes.
Oh, okay, that's it. Thank you. No, I just want to take
Got it?
picture.
Oh, I'm gonna send the PowerPoint anyway later.
Okay.
Okay, now let's talk about electron transport inhibitors. It's basic information, mostly memorization. I'll explain whatever you need to understand, but mainly the rest you just need to like memorize these, and I'll tell you what you need to understand to basically apply it in the examination. Okay, so the electron transport chain inhibitors, as from the name you can tell that basically they inhibit the transport of electrons, right? So we have three mentioned in your book. First of all is anti-myosin A, which is inhibit cytochrome B in the complex 3. And then we have amytal and retinone for complex 1. And we have carbon monoxide and azide and cyanide for complex number 4. So this is basically memorization, okay? Can you go to the next slide? Okay. So let's say, for example, what is the results that are coming as a result from like these inhibitors? Let's say, like, let's look at complex 3. We said anti-myosin, it can work on it and inhibit it, correct? Okay. So now, assuming I gave someone the anti-myosin, right? It's like an antibiotic. So imagine I gave it to someone. What is going to happen to the electron transport chain on the cycle? What is going to happen? It's gonna get inhibited, right? But more specifically, as Jasmine explained, first of all, from complex 1, we're taking the high electrons from the NAD edge, right? Which is complex 1, which is called NAD edge dehydrogenase. It's gonna take the hydrogens from the cycle that happened before. So then it's gonna transport the complex from electrons. It's going to transport the electrons from complex 1 to complex 2 to complex 3 until it reaches the end, and then we synthesize ATP. Okay, so now if we have anti-myosin given, it's going to inhibit complex 1. What it does, it's basically going to bind to the complex number 3, and it's going to prevent any electron from passing through it. So what's going to happen to everything before complex 3? It's going to have the electron on it still. So it's going to be reduced, right? And then whatever on the post the complex 3, it's going to be oxidized. Is this clear? Okay. You can go next.
Okay, so basically, we have something called the chemiosmotic theory. What the chemiosmotic theory does is it's trying to explain how the electron transfer throughout the ETC is used to generate ATP. So how is it used to generate ATP? We know that, as we basically mentioned, when electrons are transferred throughout the ETC, protons are pumped in each, like most of the, in each complex except complex 2, but protons are getting pumped from the matrix into the IMS, okay? Now, when protons are pumped from the matrix into the IMS, this creates an imbalance in both the concentration of protons, and because protons are charged, it also creates an electrical potential gradient, okay? So this is like basically it creates an electrochemical gradient, specifically called the proton motive force. Now, when we want to return these protons back into the matrix, we're going to need a protein or a transport channel. Why? Because protons are charged, so the membrane is impermeable to charged ions and therefore protons. Now, where is the transport protein found? We have two. We either you have ATP synthase, or you have something called the inorganic phosphate proton translocase importer. I forgot exactly what it's called, but we'll get to it later anyway. So these are the two channels that transport protons from the IMS into the matrix. Now, specifically, what they do, okay, is while they transport protons from the IMS into the matrix, they're also going to synthesize ATP. So that's how the electron transfer throughout the ETC generates an proton motive gradient, a proton motive force, which results in ATP synthesis. Now, what are the evidences of the chemiosmotic theory? How do we know it's actually real? We have number one, when you measure the pH of the IMS or the intermembrane space and the pH of the matrix, you'll find that the pH of the IMS is slightly more acidic. Why? Because we're pumping protons from the matrix into the IMS, so there's more protons in the IMS. Number two is that when you disrupt the inner mitochondrial membrane, ATP synthesis stops. That means that ATP synthesis occurs in the inner mitochondrial membrane, and it requires the integrity of the inner mitochondrial membrane to occur. Now, the third point is that some ATP synthesis inhibitors are specifically known to dissipate the proton gradient. Like what? We have two examples that you should know, ionophores and uncouplers. Now, the uncoupler, an example of that is dinitrophenol, okay? Which basically, what it does, it physically picks up protons from the IMS and returns them to the matrix. And ionophores, what they do, so for example, gramicidin A, it forms a channel, okay, that allows protons to move by themselves back from the IMS into the matrix. Okay, so let's...
I have a question.
Yes.
I didn't get the overall. So the protons will be from the matrix to the IMS, or from the IMS to the matrix?
Sorry?
Will the protons come from the matrix to IMS, or from the IMS to the matrix?
Protons are pumped in the complexes. In the complexes, protons are pumped from the matrix into the IMS. This creates a gradient, okay? So because you want to reduce the gradient or return it back to normal, then protons will go back from the IMS into the matrix. Do you get that now?
Okay, so in this theory is we're doing the opposite, from the IMS to the matrix.
The theory talks about both of them, both directions. Basically, in the ETC, protons are pumped from the matrix into the IMS, and then during ATP synthesis, or for ATP synthesis to occur, the protons will go back from the IMS into the matrix. Does that make sense now?
Yes.
Okay. So let's move on to the questions.
I think you're muted.
Okay, now guys, let's talk about ATP synthesis. As Yasmin explained, what we have in the inner membrane, we had like four complexes, right? Each are basically pumping hydrogen into the intermembrane space. So from the matrix, we're basically pumping hydrogen ions into the intermembrane space. Now, to synthesize, it's called, we move the electron through these to make ATP at the end. Can you get to the next? Okay. So as you see, this is exactly what's happening. So after all of these complexes pumping all this hydrogen, first of all, it's gonna become a protomotive difference, or basically like electromagnetic difference, because there's a lot of hydrogen ions in the inner membrane space and not enough inside of the matrix. Or basically it just creates a difference. So as a result to this, we can now synthesize ATP. We have a molecule also in the inner membrane, or a protein that's called ATP synthase, or also complex 5. What it does basically, it has a very unique structure. It's gonna use this difference to take all of these hydrogens in the intermembrane space and pump it really fast into the matrix again. And as a result, we're gonna have ATP synthesis. Next. Okay, so as you see to the right, this is the structure that's very unique. You have to know it because it came like 3 times total in our year. So basically, we have two structures, right? We have it's made up of two subunits. Subunit called F1 and a subunit called F0. What you need to imagine it is from down to up. So basically, the hydrogens are gonna be a lot from down, and the place that's like the head part is in the matrix. Okay. So we have the F1 unit that's made up of an alpha, beta, and gamma and epsilon subunits. And the regular one, the one F0, it's made of A, B, C subunits, okay? So I'll tell you how it functions now. Can you go to the next? So what happens exactly, we start from below, as I told you, we have a lot of hydrogen protons there, right? So then they all together very fast, go through the whole, do you see this green thing that's called the C? It will go through it It will go through the A that's on top of the C. And this is called what? It's called the rotatory subunit, or F0. So it's going to go through it, and it's going to cause the C to rotate very fast. If you see the C molecule is attached to a gamma, the one that has a circle, the white circle. This gamma thing, it's going to keep rotating as well as a result, which is underneath what? It's underneath the beta portion. Do you see it, guys? That's part of the F1. Does this make sense? Just look at the photo and it's going to make sense when you're looking at it. Okay, so it's going to go through C1, A, and it's going to cause the rotation. Going through gamma starts rotating, and then basically making differences in the beta portion. As a result, will synthesize the ATP. And you have to know that it's like the C ring is made up by C10 to 12 units. It rotates by an A unit and delivers this rotation to rotator 1 in the F1 unit. Can you go to the next? Okay. In the beta, when we're looking more closely to it, we can see that there are 3 states. As I told you, what is rotating? The gamma subunit, right? After it's rotating, it's going to sometimes squeeze things, and as a result, basically force the ADP and the phosphate to make ATP, which we need. So we have 3 states. This is also very high yield and definitely coming. So first of all, we have the loose state. This loose state, the ADP and phosphate are attached to the, as you see this one, the blue first picture. It has the ADP and the phosphate attaching to it. This is called the loose state. Both ADP and phosphate are present, but nothing too tight, just there, okay? Then after it rotates again, after what the gamma subunit is kind of pushing on it and making it tough, so it becomes into a tight state. This is the beta subunit. So then the gamma shaft, after it rotates, it's gonna create the ATP and the phosphate. It's gonna make pressure, causing them to kind of become very close together. So ADP and phosphate, which are required to make ATP, they come very close together. Then in the open state, we have ATP finally formed, and then we can release it. Next. Okay. See this yellow thing? This is called the stator. Also, came before as a question. So we have B, which we said it's from a subunit that's called F0. And we have the delta, which is, both are working as a stator. They basically hold it, stop it, until we have until the proton difference is made. After that, ATP synthesis starts. Okay, next. And now she will explain how much is made after each rotation and all.
Okay, so now that we've gone through oxidative phosphorylation as a whole and the entire process, let's talk about how we control this, and specifically, how do we control ATP synthesis? So if we want to control ATP synthesis, you know that we either control the products or the reactants. So we're either going to control ATP or ADP or PI, okay? Now, how do we control ATP and ADP and PI? So you know that ATP is basically the same as ADP, but it contains one extra phosphate group, okay? And because the phosphate group is negatively charged, that means that ATP is more negatively charged than ADP, okay? Now, remember, if we go back to the proton motive force, okay, the IMS is more positive than the matrix. Why? Because it contains more protons. Now, this is of significance in this section. Why? Because the IMS, since it's more positive, it's going to attract ATP more. And the matrix, because it's less positive, it's going to attract ADP more. Okay? So that means that ATP and the transport of ATP into the IMS and the transport of ADP into the matrix is more favored. And they will be both transported by a single transport channel also, or an antiporter called the ADP-ATP translocator. Okay? So that's for the controlling ATP and ADP like concentrations or levels in the matrix and the IMS. We use the ATP ADP translocator. Now for phosphates, okay, we have the one that I mentioned before, the inorganic phosphate proton symporter, okay, which transports the phosphate and the proton in the same direction, okay. So from the IMS into the matrix, and that's it basically. And just like what I mentioned, that basically when protons go back from the IMS into the matrix, they also go back with inorganic phosphate in the same direction, and the transport channel is called phosphate translocase. Now, in total, how many protons are transported per molecule of ATP synthesized is going to be 4. Why? Because 3 protons are used to drive the ATP synthase, like mentioned before, and the 4th one is used in this phosphate translocase symporter. Now, for you to imagine it better, look at this diagram. So you have the inner IMS here and the matrix here, okay? The IMS is more positive, so ATP will move into the IMS, and the matrix is less positive, so ADP will move into the matrix. Okay. Now, when the protons, because the IMS contains a higher concentration of protons, then protons will go through this channel to back to the matrix, and with them, inorganic phosphate will be carried back into the matrix. Now, notice that ADP and phosphates are both carried into the matrix, so then they both go through ATP synthase and they form ATP, which is transported back to IMS, and so it kind of forms a cycle. And the benefit of this is that because ATP is transported into the IMS, so then the concentration of ATP in the matrix will decrease, and so this favors this ATP synthesis by this enzyme. So hopefully you got that. Okay, now let's go back to the question. Okay, so any questions regarding this? Do you guys not get the questions, or are you fine with it?
No, we're fine with it. It's clear.
Okay, that's great.
Okay, now let's explain the creatine kinase and creatine. So to make it very simple, it's basically like an enzyme that's present in the cell. The last thing I told you guys that we synthesized ATP. Type, where did it go? It was inside the matrix, okay? So now in the matrix we have ATP. In the mitochondria, it's the main generator of ATP. We really depend on it. So there are a lot of sensors to the energy, you know. So creatine solves creatine kinase and creatine system solves an issue for us. As you see, we have mitochondrial creatine kinase, cytosolic creatine kinase, and cytosolic ATPase. These are basically we have ATP in the matrix, right? The thing is, assuming someone like is doing an exercise right now, so you need energy very fast, like for example, weightlifting. Okay, so the muscle cell, it needs energy very fast, and the way that we're transporting ATP outside of the cell to other cells that need energy is kind of slow, like let's say 10 to 20 seconds slow. So this creatine kinase and creatine solves this issue for us by basically taking the energy and transporting it out of the cell in a way that's kind of faster. So can you go to the next? Okay. As I told you guys, one more. Okay. We have a molecule that's called phosphocreatine. Okay. What happens from the creatine molecule itself? It's gonna go to the matrix. It's gonna go take the phosphate and go outside and give it to the cells, because all of the cells have like an ATP in them. When the ATP is done, it's gonna get converted into an ADP. That's how we get energy, by breaking this ATP itself into a phosphate and an ADP. So while taking this creatine and taking from the inside of the cell, it's going to take a phosphate group with it, and it's going to go to the muscle cell, for example. Now when it goes, it's going to give its phosphate back again to the ATP. And now we have like unlimited source of ATP, you know? Next. Just also know that we have an isozymes with it, and that's like all you need to know about the creatine kinase. Okay, I have a question here. You can answer in the chat as well. Which enzyme is found specifically in the intermembrane space and changes creatine into phosphocreatine? Exactly, yes, mitochondrial creatine kinase. Okay, next. How does the empty creatine alert the mitochondria to increase the energy production? Does it diffuse back to the mitochondria and act as a signal for higher demand? It blocks the ATP synthase to save energy. It turns into lactic acid to provide an alternative fuel. This is a next question, guys. Or it binds to oxygen to speed up the electron transport chain. Which one is it? How does creatine alert the mitochondria that we need more energy? صح, exactly. Excellent, Abdullah. Okay, next. It diffuses back, as I said, and basically takes more ATP and goes back again. Takes a phosphate and gives it to the ADP cells.
Okay, so sorry. Oh my God. Oh, okay. So just to recap everything that we did so far, we basically have 4 complexes. Complex 1 received electrons from NADH and donated them to ubiquinone. It pumped 4 protons in the process. Complex 2 received electrons from FADH2 and donated them to ubiquinone and did not pump any protons in the process. complex 3 received each ubiquinone or each dihydroubiquinone went to complex 3 to donate the electrons to cytochrome C, forming one Q cycle, okay? And it pumped 4 protons in the process. Now, complex 4, for every Q cycle, it pumped only 2 protons. Why? Because it requires 4 cytochrome Cs to pump to produce water, but each Q cycle produces only 2 cytochrome Cs. Type. What does this mean? In each NADH, we already covered this in the questions that we went through, but each NADH is going to pump 4 protons in the complex 1, 4 protons in complex 3, and 2 protons in complex 4. That results in 10 protons getting pumped. And so since each ATP synthesized requires 4 protons to, you can say that 4 protons are required to produce each molecule of ATP. So, and we have 10, so we get 10 divided by 4, so 2.5 molecules of ATP produced by each NADH. Now, each FADH2 that enters complex 2 will not pump protons here in complex 2. It will bypass complex 1, so no protons pumped here. It will only pump 4 protons per complex 3 and 2 protons per complex 4. So that's 4 plus 2, 6 protons pumped per FADH2. And that means that each FADH2 will produce 6 divided by 4, which is 1.5 molecules of ATP. Is that clear?
Yes.
Okay. So now, but then we have a problem with the NADH one. Okay. What did we say that are sources of NADH? We can have fatty acid oxidation, amino acid oxidation. Most oxidation produces NADH, TCA cycle, and also glycolysis. But where does glycolysis occur? Glycolysis occurs in the cytoplasm. Now, the problem is that, okay, the outer mitochondrial membrane is permeable to anything, but the inner mitochondrial membrane is not permeable to NADH. So NADH produced in glycolysis, which occurs in the cytoplasm, cannot enter the ETC. Why? Because it cannot pass through the inner mitochondrial membrane. So how do we bypass this problem? We can send we can give NADH electrons to a carrier that can cross the inner mitochondrial membrane. Like what? We have 2 shuttles, which are basically 2 methods by which we do this. We have the glycerate 3-phosphate shuttle and the malate aspartate shuttle. So in the glycerate 3-phosphate shuttle, what happens is that we can give NADH electrons to DHAP, okay, converting it to glycerol 3-phosphate. Now, glycerol 3-phosphate is permeable in the inner mitochondrial membrane, so it can enter. And here, just like I mentioned before, the glycerol 3-phosphate dehydrogenase, remember that enzyme? So there's another one in the inner mitochondrial membrane. And basically, what happens is that glycerol 3-phosphate will be oxidized back to DHAP, and it will donate its electrons to FADH2. FAD, sorry. So even though you should know that even though that these two are the same enzymes, glycerol 3-phosphate dehydrogenase, they're the same enzyme, but the one in the cytoplasm uses NAD, and the one in the inner mitochondrial membrane uses FAD. Okay? Now, FADH2 will donate the electrons to ubiquinone, and this process will bypass both complex 1 and complex 2. So, only 4 protons will be pumped per by complex 3 and 2 by complex 4, so a total of 6 protons pumped. That means that each NADH that enters this shuttle will produce only 1.5 molecules of ATP. That makes sense? Okay. So
Can
now,
you
sorry.
repeat that, please? Last part.
Okay. Basically, in we have so one of the carriers that we can give electrons of NADH to is DHAP. We can give it to DHAP, forming glycerol 3-phosphate. Okay, and the enzyme that catalyzes this reaction is called glycerol 3-phosphate dehydrogenase. Now, glycerol 3-phosphate is permeable, so it can enter the it can enter the intermembrane space, and it will basically get oxidized back to DHAP by the same enzyme, glycerol 3-phosphate dehydrogenase, that is found in the inner mitochondrial membrane. But the difference is, even though they're the same enzyme, the one in the mitochondria uses FAD, while the one in the cytoplasm uses NAD. Now, FAD will be reduced to FADH2, which can donate the electrons to ubiquinone. Now, because this process bypasses complex 1, then we can only pump 4 protons from bicomplex 3 and 2 protons by complex 4. So that's a total of 6 protons pumped across the inner mitochondrial membrane, and therefore each NADH molecule that enters this shuttle will produce 1.5 molecules of ATP. Does that make sense now?
Yeah.
Okay. Sorry. Now, the second shuttle is called the malate aspartate shuttle. Now, it looks really complicated, but we can simplify it. So basically, we can donate the electrons of NADH to oxaloacetate, okay? And this will convert it to malate. Remember, this was a step in the TTA cycle, but it was the opposite. It was malate to oxaloacetate, but it's the same enzyme. So, so malate will now enter the matrix, okay, and it will form. Now, sorry, oh my god. Oxaloacetate will be converted to malate. Malate will enter the matrix through a certain transport protein called the malate alpha-ketoglutarate transport protein. And this is an antiporter. It takes in malate into the matrix, and it also transports alpha-ketoglutarate out of the matrix, okay? Now, malate in the matrix will be converted back to oxaloacetate, and it will donate its electrons to NAD. Now, because we're donating the electrons to NAD, we're not bypassing complex 1. So we can pump 4 protons through complex 1, and 4 through complex 3, and 2 through complex 4, which is a total of 10 protons pumped across the inner mitochondrial membrane. So this process does not bypass complex 1, whereas the other one does, because FAD is the acceptor, not NAD. Now, oxaloacetate is impermeable to the membrane. Okay, see here, it didn't even cross the membrane when it was converted. It has to be converted to malate to cross the membrane. But oxaloacetate is impermeable, so it has to be converted to aspartate, okay? And now aspartate will go back to the intermembrane space through another antiport called the glutamate aspartate antiport, I believe. Yeah, glutamate aspartate transport protein, which is an antiport that takes in glutamate into the matrix. This is a summary of all the ATP that can be produced by the different shuttles of glycolysis and by the link reaction in the TCA cycle. So it's the same as the red table, but less simplified. So the glycerol 3-phosphate shuttle will produce a total of 30 molecules of ATP, and the malate aspartate will produce 32. Okay? Now, let's go to the questions, if you don't have any questions.
And can you repeat it? Can you go back to the table?
Sorry?
Can you go back to the table?
This one?
Okay.
Is that it?
This is I just took a picture.
Oh, okay. Okay, congratulations, guys. Last question. But do you guys get the previous questions that we did? If you don't get it, let me know so that we can go back. Okay, let's see who are the total winners of the Kahoot since we finished. So in third place, we have R. And then second place, we have K. And then the first place, we have Abdullah. Okay, so congratulations, guys. And congratulations to the fifth place. Okay. Did you guys get the Pacademia so far? We're almost finished.
طيب يلا إن شاء الله guys يعني ما حنطول عليكم hopefully 10 minutes in 10 minutes إن شاء الله okay. So now let's talk about uncoupled electron transport okay. We said, do you see this diagram? This is the ATP synthase. What's the thing that's kind of working on and basically synthesizing the ATP? That I have a difference لأنه because we have a difference because we have a lot of hydrogens that were pumped into the inner membrane space. That's the reason that's causing me to get energy. What uncoupling does, like multiple molecules, or for example, when in babies, they have like a certain type of mitochondria that's always kind of doing uncoupling because it generates heat. So in the uncoupling, do you see the arrows, these red arrows? What happens is that there's a hole in like throughout the inner membrane space, so preventing the difference from happening, and then like the ATP synthase complex using it to make ATP. This is all you need to know. You need to know also that we have UCP2, UCP3, يعني multiple ones, okay? Now let's talk about like reactive oxygen species. As Jasmine said, first of all, oxygen is a very like reactive molecule. It's a diradical. That's why it's very easily like it can become reactive. Now, in the mitochondria and in the cell, when our bodies are made up of multiple cells, these reactive molecules, they can bind to lipids, they can like interact with proteins and prevent them from like working properly, so it can ruin a lot of things. That's what's called reactive oxygen species. It's something called hydrogen peroxide, which is H2O2. This is a reactive species, but at the same time, it's more of a lighter part, and we have a superoxide radical. Okay, we have many systems in our body and basically in the mitochondria itself that's working on to kind of prevent these damages from happening as much as possible. Not only them, we also have other mechanisms, but we need to know this for like our exam, okay? So we have, do you see this NNT? This is a very important. It's basically getting the NAD pH inside, and I'll explain all of these in detail, okay? We have 4 enzymes that we need to know about and 2 systems that are keeping the enzyme going. So we have superoxide dismutase, this enzyme number 1. We have glutathione peroxide, we have peroxidines, we have catalase, okay? So first of all, let's talk about the primary antioxidants enzymes that we talked about. Now we have what? Superoxide dismutase. First of all, what's a superoxide? Do you see this 2O with the charge? This is the superoxide. Now, this is very dangerous because you see there's an electron that's free on it. So it can literally attack anything. And remember, we are working with the mitochondria. So if something gets ruined there, يعني it's much harder and more like side effects that are going to happen. So that's why we have something called superoxide dismutase. Its job is to take this superoxide, get it to become into a peroxide, into H2O2, as you're seeing in the equation. Okay, we have 3 forms, SOD1, SOD2, and know each one like has the first one and the third, they have copper and zinc. The 2, it's located into the mitochondrial matrix and it has mannose in it. MN group, not mannose, sorry, MN group, okay? The second system that we talk about is called the glutathione peroxidase. What was formed in the first one? We said we got the superoxide into a peroxide, right? Which is much simpler, اللي هو H2O2. So we use this glutathione system to kind of use this to convert peroxide, which is H2O2, to water, which is, of course, it's not toxic at all, okay? Water. So this is showing us the equation that we have it as it's called glutathione peroxidase because it's gonna work on converting the peroxide into water. Next, know the forms also 1 and 4. Okay. Again, we have catalase. This came in a question actually. I believe like it was catalase is a system found where mainly it's found in peroxisomes. If you studied cells, you know that we have something called peroxisomes in cells. The peroxisomes we have mainly catalase. Okay, it's a heme-containing enzyme. It converts two molecules of H2O2 into water and oxygen. Very similar to what glutathione peroxidase. They're all working very similar to each other. Okay, we have also something called PRX or peroxypyridoxines. Okay, it's also thiol-containing enzymes and that detoxify peroxidants as well. So all of them are working on peroxides and converting it to water, while the only one that's working in superoxide, اللي هو the first one, superoxide dismutase. Okay. Next. Now to keep these, yes.
Can you repeat the last thing, like literally the last sentence you said?
Go back.
That all of them do something except the first one, the one that works with the superoxide.
We said a superoxide molecule is what is hydrogen literally with an electron on it. Okay, the rest after it works with, it's called what the enzyme called superoxide dismutase or SOD. SOD is going to take this molecule and convert it into a molecule called peroxide. Peroxide is H2O2. So when we have this, it's slightly less like hostile than the superoxide molecule. So all of them are using this peroxide and converting it into water. That's all. So the first one is dealing with the most severe one, which is converting the superoxide into peroxide, and then all of the other three enzymes, they use this peroxide, H2O2, and يعني make it into water, which is not harder. Do you get it now?
Yes.
Okay. Next. طيب, now we have also two systems. يعني احنا we said we have enzymes. We have two systems that are continuously making these enzymes be able to work. Okay, we have GSH or reduced glutathione, and it's basically a tripeptide. So we have two enzymes that are working in this system. احنا our main thing that we're using, it's called glutathione. Glutathione in natural form, it doesn't have a hydrogen group. Okay, what do we use? Remember the NNT I told you about? It brings a NAD pH. It's gonna take the hydrogen from it. It's gonna take the hydrogen from it and put it on the glutathione itself. After that, we're gonna get 2GSH, which is what? Reduced glutathione. When we have a hydrogen, we know it's reduced, and we don't have it, it's oxidized. Okay, why is it reduced? احنا we said it works on what? Peroxide into water. So imagine you have, يعني I don't know if I can't point, but do you see the H2O2 on the left here? Yes. So this one, we're gonna use the GSH. It has hydrogens. Okay, so imagine we're putting two hydrogens with this together. هيعطيني 2 water, because the water has what? It has 2 H's and 1 O. So if I'm using, imagine like we have H2O2, which is called what? Peroxide. We're gonna use this peroxide, نعطيه another hydrogen. خلاص, I got 2 water now. Is this clear?
Yes.
Okay. Next, the other system that's helping us, يعني don't mind the notes, it was شوي confusing, but it's easy in general. Okay, I'll tell you the name. Let's start from the left. We said this is working with the radicals, right? This system is different. The thioredoxin, from its name, it works with thiol groups. As we know, we have a lot of proteins that have or two proteins that have thiol groups, right? And also in the body, in the cells, we have groups that are dependent on the thiols. So as you see here in the left, let's start with the left. We have a protein with an S group. using the thyrodoxine reductase, TR. I wrote all of the names so you guys can like keep up. So we have first of all TR. TR standing for what? It's thyrodoxine reductase. Okay, it's gonna basically, the natural form is SH2. So it does what? It returns it to its natural form. So it takes any radical. مثلا if I take a thiol group and remove the hydrogen from it, we're gonna have an S only, which is very reactive. It can attack cells. It can cause damage. So that's why we need this system. So we're gonna use thyrodoxine reductase. From its name, it's literally gonna like reduce the thiol groups. It's gonna return the thiol group into life. Okay, so we have a protein with an S, as you see on the left on the top. بعدين after using thyrodoxine reductase, it's gonna make it natural again, protein S2. طيب, once the thyrodoxine reductase, it had an H by itself. It gave its hydrogen to the protein, so it becomes normal again. So as الحين I need something to regenerate the thyrodoxine reductase. Okay, so now to use this, we're gonna use a thyrodoxine. Okay, thyrodoxine, all of them, they're basically gonna use the like kind of take the hydrogen, give it to someone else, and then they need their hydrogen back again. So thyrodoxine is gonna use what? It's gonna use the thyrodoxine reductase. Okay, from the name. So in the middle, like the closer to the number 2, this is thyrodoxine. Okay, by thyrodoxine reductase, it's gonna get its hydrogen back, and we said it does what? It gives the protein its hydrogen again and making it into normal. It's not reactive anymore. طيب, once the thyrodoxine reductase itself, it gave to the thyrodoxine, how can I return the hydrogen again? By using NAD pH. Is it complicated? Guys, did you get it?
Maybe.
Okay. طيب, we can start from the very complicated. Okay. طيب. Now, let's start from the right. Aisha, I need you to know 3 names. Okay, we have a thyrodoxine. Okay, it's a small protein. We have thyrodoxine reductase. From its name, it's gonna deal with the thyrodoxine, and it's gonna reduce it. Reductase. Okay, it's gonna give it a hydrogen group. Okay, and we have the peroxyreducing enzyme, which deals with like the hydrogen groups as well, but like more with the peroxides. Okay, directly. We said the thyroreducin centered system, this system as a whole, its main job is to deal with preventing proteins from attacking protein radicals. Assuming now we have a protein with an SH group, okay, a thiol group. If this thiol group, let's say it like lost a hydrogen and became a single S, this is very harmful. Is this clear so far?
Yes.
طيب. Now all of the process is literally hydrogen. They're taking hydrogen and giving it to each other. My main goal, look at the left, protein S is follow the arrow, it's going to convert into protein SH2. Please, Abdullah and Maryam, I think, just follow the arrow. Okay, we have protein S2, it became into protein SH2. How? We used thyroreducin. Okay, point at the arrow next to it, TRX SH2. Follow the arrow, صار S alone. It doesn't have an H. This is what reduction is. It gave its hydrogen to the protein to make it normal again, to make it into SH, the normal form. طيب? Okay, now when we use the TRX first, to memorize it like this, we have TRX first, okay? It gave the hydrogen to the protein. Now I have a normal protein, but thyroreducin itself is not normal, okay? Do you see where the arrow is pointing? It's as literally empty. So now I need to make it back into SH so that it becomes like normal again. Okay, I need the H again. مين راح استخدم thioreducin reductase, literally using the same يعني نفس الشيء TRX reductase. يعني it's gonna use thioreducin and put a hydrogen on it, reducing it. Now لما رجعته أنا into SH, I can use it again. خلاص, a lot of proteins can come and we convert it into the normal form again. Do you guys get this point so far?
Yes.
Okay. طيب, we said TRX first, okay? TRX dealt أو thioreducin dealt with the protein, حولناه طبيعي. I don't have the hydrogen for it, I used thioreducin reductase. So we took thioreductase in hydrogen وعطيناه to become the normal. Do you see the label normal? Okay, so صار عندنا now thioreducin normal again on the top, the SH again. Now, after I gave it on the top, we see TRS2. This TRS2 is not normal. Okay, I need its hydrogen again. Who am I gonna use? Do you see next to it exactly? Not PHH. هناخد هذا ال H again and regenerate thyroreducin reductase again وبس, it keeps going. خلاص, clear now?
Very clear. Yes.
Okay. خلاص, next. الحمد لله. If you guys need me to repeat, it's fine. طيب, this is exactly just what I explained. It's the same point. So if anyone has any questions, you can write on the chat or ask. Okay, يلا next. Okay, guys, this is now like for all the part I explained, just because I want you to like use your memory more. Okay. طيب, you can answer in the chat. This has two answers actually. Which of the following remains oxidized when the electron transport chain antimycin is added to a suspension to actively respirating mitochondria? يعني we have a mitochondria. Basically, we give antimycin. Antimycin works on what? Works on complex 3, صح? So we have it, it's gonna be attached to complex 3. طيب, now that we have something blocking complex 3, what's going to happen before the complex? What's going to happen after the complex? Abdullah, can you answer?
Ubiquinone will stay UQH2. It will not be reduced.
No, it will stay reduced.
Yeah. I mean, it will not go
reduced.
back to oxidize. It will not give its proton.
صح. Okay. What about cytochrome A? It's part of what?
It's part of the complex 3.
No, it's part of complex 4.
Oh.
So it's gonna be reduced or oxidized?
Oxidized.
صح, because what? Everything, the electrons before the complex 3 are blocked. Post of it, it's عادي, it's transporting and everything is fine. Okay? Cytochrome, yes.
Cytochrome C will also be oxidized because it's not going to get reduced by ubiquinone H2.
No, but antimycin is blocking complex 3. So cytochrome C is before complex 3.
Oh.
No, cytochrome C is after complex 3.
Oh, that's what I was thinking.
I'm sorry. Yes.
هما اثنيناتهم صح.
Yeah, I said it's two answers.
Yeah. So two answers correct, like she said.
So the answer is what? Who remains oxidized?
A and C.
A and C, yes. Yes, صح. Okay. طيب, which of the following ATP synthase components prevent the alpha-beta hexamer from rotating? If you remember the picture, guys, of how is the shape, how is everything organized, we said two things are kind of stator or holding things from moving? What are they? Which this
B
is one
and
answer.
gamma.
What?
B.
صح exactly. B and not gamma. It was a different delta. Beta and delta.
Yeah, I mean delta, sorry.
Can you go next slide, Yasmin? So here, do you see this thing, the on top, the yellow thing? All of it is holding the thing from rotating. We have the delta and B. So don't get the letters mixed عشان whatever you kind of were like not sure about this session, يعني make sure to revise it extra hard عشان you don't do the same mistakes. طيب يلا the next. Okay. Which component of the ATP synthase is responsible for the catalytic conversion of ATP and phosphate into ATP? We talked very detailed. We were like, it rotates and it's gonna يعني بيصير loose. We have tight. We have هذا. What were we talking about?
Searing, which rotates.
Delta.
No, rotate. اقرأ the question. Read the question. Conversion from ADP and phosphate into ATP. Searing does rotate, but who's converting it into ATP?
Delta Sabina.
What?
Delta.
It's beta, صح. Exactly. Beta subunit, we said we have three, the loose and tight and all of these were part of the beta subunit. Okay? Next. طيب. This is the binding change mechanism. What happens when a beta subunit is in the tight state? Does ATP get released into the mitochondrial matrix, or the subunit is empty and ready to accept new substrate, or mechanical energy forces ADP and phosphate to form a covalent bond, ولا ADP and phosphate are loosely bound and can still leave the site? What's your answer, guys?
C.
صح. So because it's tight, it's gonna kind of force them to become, and then when it's loose, we can release things. طيب. Next. I think maybe this the last. خلاص احنا we're done, guys. أصلا these are just questions for revision. Okay, this is the last question. طيب, the ADP and ATP translocator is described as an antiport. What is the correct exchange it performs? We talked about the movement of ATP. We said when ATP is synthesized, it's going to go to the matrix, right? And at the same time, يعني to
I have
get
a
it.
question.
Yes.
You said the inner membrane space will take more ATP because it's positive. So it will be synthesized to the matrix?
Say the question again.
You said that the inner membrane space is negative. So it will take قصدي it's positive, so it will take ATP. So when you synthesize ATP, it will go to the matrix?
Yeah, صح. It will go to the matrix, yes. But بعدين we need to take it to cells. So that's why we did what we got the creatine shuttle, and it helped us to get things out, you know, to get it to outer cells. يعني here now بماتريكس الميتوكondria we needed to like for other cells in the body, and that's all. طيب antiporter معناته something goes in and something goes out. So what do you guys think the answer is?
Moves ATP out of the matrix and ADP into the matrix.
Okay, excellent. Thank you. شكرا guys, خلصنا. والله sorry تأخرنا عليكم, and please يعني forgive our shortcomings if we didn't explain anything properly, and feel free to like text us if you need any help. We tried to do all of the high yield stuff. يعني anything we explained is inshallah يعني coming. The other things يعني هيك just side details, not that important. والله يوفقكم.
أبدا, thank you, ما قصرتوا.
Guys,
was
thank
very
you so
good.
much.
Very nice.
Okay, I hope you understood everything. Do you have any other questions you would like to ask us?
No, thank you.
Okay, great. And if you do have any questions, you can also feel free to text me or Aisha on WhatsApp. Okay? Thank you so much.
I have a
guys.
question.
Yes.
The equations in each enzyme وهي we should know that.
أنتي listen ترى هم they're there to help you, you know. So أنتي try to understand them. If you didn't get يعني try to see the recording and write notes next to it. If you still didn't get it يعني I'll re-explain it again. بس هم they're gonna help you. يعني I don't remember exactly the إنه coming in the exam. بس knowing the names it's gonna help you a lot يعني. مش maybe like not all of them didn't come يعني mostly it's high yield topic in general, the antioxidant system, you know, it's very like it has a lot of information but it's high yield at the same time يعني I made it as simple as possible. فأنت try to see the recording if it's you still didn't get it or you want me to explain it again, no issue يعني. بس yes you need to know يعني افهميهم أحسن.
Okay, thank you.
Is that it for everybody? Okay, yalla. See you guys, and thank you so much for attending. Thank you so much. You guys did awesome. Or you participated so much, and we're so like happy. And hopefully, inshallah, that you guys are also happy and satisfied with our explanation. Obviously, like I said again, if you guys don't understand anything, you can feel free to text us at any time. Or inshallah, we can help you. Okay.